Good morning, friends. Myself Sudhanshu. Welcome to Conceptual Classes. In this my new videos, we'll discuss some of the topics of plant breeding. Plant breeding nowadays, very well that it is one of the important topics or important parts of any state service exam or a Union Public Service Commission exam. Any service commission exam, this is one of the most important topics, and which questions are asked uh, from different chapters. So I have prepared this course in such a way that each and every chapters, whatever required for various competitive exams should be covered. I cannot prepare all these chapters in single videos. That's why I have prepared these videos in parts. First, each video there will be 3 to 4 chapters depending upon the length and breadth of the chapters. And these chapters we will discuss slide wise. So in this video, we'll discuss some introductions about plant breeding, what is plant breeding, why we are studying plant breeding, what are the objectives of the plant breeding, what are the activities involved in the plant breeding, what plant breeders do for release of the varieties, what are the important achievements in plant breeding, what are the undesirable consequences involved in the plant breeding methods or traditional breeding methods, what are domestication, what are germplasm conservations, and what is the center of origins. So these topics will be covered in this video. Objective of the course is quite clear that the main objective of this course is to clear the concept related to plant breeding, genetics and agriculture, which are mainly important for various exams without attending any coaching tuitions in minimum time and efficient utilization of your time as per your convenience. Because if you join coaching, you have to attend the class as per the convenience of the as per the schedule of the coaching and tuition. But this video has no any restriction. Whenever you got free time, you can watch this video. Target audience may be UPSC, state PSC, BPSC, particularly state agriculture officer exams, BSc agri students which are studying in first year to fourth year. Botany honors students also got benefited. Please don't confuse that only watching video will be sufficient to score maximum exams. It will only help you to clear the concept, systematize your knowledge, and selective understanding and easy recall of the topics. Please share, like, comments, and subscribe the video if it really helping you. Or if you are disliking the video, please give the comments why you are disliking so that I can improve the same in my next video. If you have any doubt or confusion or you want any new videos on a specific topic, please write the comment box given below. It is my personal guarantee that video will be uploaded within 24 hours. For more videos, you can search on my YouTube channel, New Conceptual Classes. So let us start. First, what is plant breeding? For agri students, it is very easy terms. It is an art, science, as well as sophisticated technology of changing the genetic makeup of plants in order to produce desired characteristics. Means whatever plants we are seeing here and there or near about us, we have to change the genetic makeup of the plant. But how we change? What is the purpose of changing that genetic makeup? The purpose of changing that genetic makeup is only to produce desired characteristics, or we can say for human welfare. Whatever changing in the genetic makeup of the plants has been done by the plant breeders, that have some objective, that have some goal, and that goal is just to for human welfare. Whatever humans need is there, for that need we are changing the genetic makeup of the plants. So it involves science as well as art as well as some technology. Means it is the combination of art, science as well as technology. So everybody is knowing there are three types of crops found in our nature: self-pollinated crops, cross-pollinated crops, asexually propagated crops. What is sex-pollinated crops? The crops of the plants in which anther of the pollen grain of the flower falls on the stigma of the same flower. It is called self-pollinated crops. If pollen of the male flower falls on the stigma of the different plants or different flowers, then it is called cross-pollinated crops. Asexually propagated crops means in which propagation of the plant can be done by some different parts of the plant like root, stem, leaves, and so on so on. So there are so many breeding methods which can be utilized differentially in self-pollinated, cross-pollinated, and asexually propagated crops. First, we are talking about self-pollinated crops. In self-pollinated crops, we are using plant introduction, pure line selection, mass selection, pedigree selection, bulk method, single seed descent method, backcross method, heterosis breeding, mutation breeding, polyploidy breeding, distant hybridization, and transgenic breeding. These are the for the improvement of the self-pollinated crops. Some techniques like recurrent selection, disruptive selection, diallele selectivity, biparental mating, or these techniques are used for population improvement. If we are improving the population of the self-pollinated crop, we are using these four techniques: recurrent selection, disruptive selection, diallele selective mating, and biparental mating. So these are the some of the techniques to improve the characters of the self-pollinated crops. What are these techniques? We will discuss in next video. Then cross-pollinated crops, we are using simply plant introduction. Plant introduction is common for self-cross as well as asexually propagated crops. Mass and progeny selections, which is only for cross-pollinated crops. Heterosis breeding exclusively for cross-pollinated and asexually propagated crops. Then composite breeding, polyploid breeding, distant hybridization, transgenic breeding. So these are the some techniques which are exclusively used for improvement of the cross-pollinated crops. Some techniques like recurrent selection, disruptive selection, and biparental mating used if in cross-pollinated crop for improvement of the whole populations. So these are the breeding techniques of cross-pollinated crops. In the next video, we'll discuss this all the topics chapter wise in detail. Asexually propagated crops, we are using plant introduction, clonal selection, mass selection, heterosis breeding, mutation breeding, polyploidy breeding, distant hybridization, transgenic breedings. These are the breeding techniques used in the asexually propagated crops. So there are so many topics, so many terms which I think you are unaware or not aware about that. Please don't take pen. In next video, whenever we discuss each chapter's detail, it will be very clear. Just here I have shortlisted or I have made this data so that in objective question you cannot confuse. Sometimes they are asking in heterosis breeding is used for which crop: self-pollinated crop, cross-pollinated crop, asexually propagated crops, both B and C, like that. So you can take out easily. This table is only used for objective solving objective questions. Now, second, what is the objective of plant breeding? Why this plant breeding chapters books came? Why we are reading this plant breeding? What is this use? So mainly the use of this plant breeding is high yield, improve quality, disease and insect resistance, change the maturity duration of the crops, modification in agronomic characteristics like low vegetative growth, high reproductive growth, like that, synchronous maturity, determinate growth, elimination of toxic substance, moisture stress, and salt tolerance, and so on, so on. So there are so many characteristics of the plant by which yield has been affected, their quality has been affected. There are so many insect and disease affecting many crops plants. So this all characters of the plant can be improved only by the plant breeding. So main objective of the plant breeding is this one only. We have to improve the yield of, example, if you are seeing the maize, last year before green revolution its yield was very less, but nowadays maize is one of the high yielding crop. Quality also we are improving in wheat, rice, barley. Disease and insect resistance variety we are releasing in wheat, maize, and so on, so on. Change in maturity duration also we have done. So these are the main objective of the plant breeding. Next class if you go, then there will be specialties: one for quality breeding, one for quantitative breeding, one for yield improvement, like that. Then what are the activities involved in the plant breeding? As you know, main objective or main activities in the plant breeding or main goal of the plant breeding is to create the variation, because unless and until no variation is there, we cannot improve the characteristics of any plant. So entire activities of the plant breeding is centered about creation of variations. So how this variation can be created? Because without variation we cannot select any good plants. First we have to create variation, then we have to do selection. After selections, after selecting best plant, we have to do evaluations. We are doing evaluation means we are evaluating what are the status of the plant on different parameters like yield, qualities, disease insect resistance, maturity duration, and this toxic substance, moisture, stress, soil tones. So we are evaluating all the parameters of selected plant. After that, if we find that whatever evaluation we have done that is found fit for the cultivation by the farmer, then we go for multiplication. After multiplication, we are doing distribution of the seeds. These are the activities we are using in plant breeding. So how variation can be created? There are two types of techniques. One is naturally existing variability and second is creation of new variabilities. Whatever variability existed in nature, we can utilize by three methods like domestication, conservation or germplasm collections, plant introductions. These are the methods to utilize the naturally existing variabilities. What is introduction, what is germplasm collection or conservation, what is plant introduction, we will discuss later. Then some new variability we are creating by that is called plant breeding techniques. It may be hybridization, it may be mutation, or you can say induced mutation, polyploidy, soma variations, and genetic engineering. So these are the some of the artificial techniques which are utilized in the plant breeding for creation of variation. Means this naturally existing variability and this one artificially induced techniques, we are the main purpose of these techniques are only to create the variations. Once variation created, we will do selections. After selection, we will evaluate the selected plant, then we will go for multiplication and distribution. These are the activities involved in the plant breeding. So what are the achievements? Next topic is what are the whatever achievement has been done in plant breeding are production of dwarf and semi-dwarf cereal varieties. As you know, everybody is knowing that Green Revolution came in 1963. Dr. Annie Warlock used Japanese variety that is called Norin 10. Norin 10 is the dwarfing gene of the wheat as a source of dwarfing gene. It was exported from CIMIT Central Institute for Research Institute for Research in Maize and Wheat. It has been situated in Mexico. In 1963, ICR has introduced some dwarf selection of from CIMIT and some variety like Kalyan Sona, Sonalika. They were exported and selected from this dwarf variety and released as a variety. That's why this green revolution came in India. So for wheat, this gene Norinten variety Kalyan Sona and Sonalika. For rice, the gene was DGO that is developed at Taiwan called Taichung Native 1 TN1 and IR8. These are the two varieties of rice which contains DGO and exported from imported from Philippines, and both are introduced in India in 1966. If you see the example of sugarcane, in sugarcane also C.A. Barber and Venkatraman at Sugarcane Breeding Institute Coimbatore, they transfer thick stem, high sugar content and other desirable characters from noble cane to Indian canes, and commonly referred as novelization of Indian canes, because earlier in India only wild cane variety were cultivated, but nowadays after novelization we are getting higher sugar content in our sugarcane. Then development of hybrid varieties, if you see the example of maize, there are so many Ganga series variety like Ganga Safed, African tall, Manjari, because these are the recent varieties of maize which has been introduced by plant breeding method. Then sorghum, you can see composite sorghum hybrid 1 2 3 4 5 6 7 8 up to 15. We have already released Bajra, you see, you can see WCC 75, PHB 10, ICTP 8203, Sarda and Savuri. In cotton also you can see H4 variety Lakshmi, Savitri, NH44, Jayalakshmi. So these are the some of the important varieties which have been used by the techniques of hybridization and plant breeding methods. So these are the some of the achievements till now in our plant breeding world. Then what happens if we are following the techniques of plant breeding, there may be some undesirable consequences also. What are these undesirable consequences? This may be genetic erosion. Narrow genetic base increases susceptibility to minor disease. So what is genetic erosions? Suppose we are doing selections or we are doing hybridization. So in the process of selections, what we are doing, we are selecting the best high yielding varieties. We are selecting the quality, the varieties which have good quality of the fat, protein, and whatever our objective or goal is. So in the meantime, the selection of the selection process, what's going on? The varieties which has some low quality or low yield, but have some specific quality trait which we are ignoring, and day by day that variety became outdated or became extinct from our natural population. So what this is the demerit of selection or plant breeding techniques. Day by day we are concentrating only on yield, quality and genetic resistance, means very limited character. We are ignoring whatever some important genes present in our wild species, and that wild species day by day got extincted. So this is one of the undesirable consequences of the plant breeding methods. Narrow genetic basis also, we are selecting only high yielding varieties, and in which genetic base is very low, poor or very narrow. What happened after three or four generations? Some new varieties or new strains of pest has been developed, and that pest or in disease and destroy all varieties. So these are the some of the undesirable consequences of the plant breeding methods. Then we are discussing the second chapter of this plant breeding, that is domestication and germplasm conservation. What is domestication? Domestication is the process of bringing wild species under human management and continued since more than 10,000 years ago. आज से 10 साल पहले से we are doing this domestications knowingly or unknowingly. So domestication can be done by two techniques. One by natural selection and second by artificial selection. Natural selection means nature also selecting some of the plants or animals. What are the factors of the nature which select the plants or animals? That may be climatic factor, maybe soil factor, that is edaphic factor, maybe biological factors, maybe natural and physical factors. What are the climatic factors? Suppose just in some area there is high rain, some area there is desert, some area cold temperature, some area high temperature. So in cold area only uh, cold this cold tolerant crop can be selected naturally. In desert area where this all water conserving plant can be selected automatically. Some soil factor also, like there are many types of soils: arid soil, this loamy soil, black soil. So depending upon soil type also plant variety differs. Biological factors, maybe pest attack, disease attack. There are so many insect, pest, weeds which are this infesting the plants. So depending upon their resistance, plants can be selected. Natural factors, these are the factors which can be selected naturally. Artificial selection means our present day crops, whatever crops we are cultivating like rice, wheat, maize, bajra, these are artificially selected by humans, knowingly or unknowingly. Selection may be directional, may be stabilizing, or may be disruptive. There are three types of selection: directional selection, stabilizing selection, and disruptive selection. What is directional selection? When we are doing selections for extreme phenotype. Extreme phenotype means, suppose we are selecting for viability, we have to select the seeds which have maximum viability. If we are selecting for the yield, we have to select the characters, the seeds which have highest yield. Means if we are selecting any extreme phenotype of a plant, then it is called directional selections. The purpose of this directional selection is just to check the viability, fertility, yield of the plants. Second is stabilizing selections, means we will not go for extreme phenotype. We will go for only intermediate phenotype, like flowering time, grain maturity. If we are selecting, we cannot select the flower or plants which flowering time is earliest or highest or latest. We will select only the plants with flowering time just intermediate. We will select only the plants which grain maturity is intermediate so that we can harvest synchronously. Then disruptive selection means directional selection in distinct ecological niche. Niche means we are doing directional selection just like we are selecting for extreme phenotype, but condition is that the selection process is going on in distinct some ecological niches. Some habitat is different, so we can say natural selection plus artificial selection both give rise to evolutions. So during the process of evolution, what happened? There may be Mendelian variations, maybe interspecific hybridization, maybe polyploidy. Now, what is germplasm? Germplasm is the sum total of hereditary materials of any plants. It may be what is hereditary materials? It may be alleles of various genes present in any species of its and its wild relatives. It may be land races, may be obsolete variety, may be varieties in cultivation, may be breeding lines, may be wild forms and wild relatives. So these are the some types of the jump are. What are land races? Land races means whatever varieties, whatever subspecies found of a particular crop species, that is called land races. Obsolete varieties means the variety which were earlier cultivated but nowadays became outdated. There are so many varieties of rice wheat which came in 1986, 1965 that were cultivated, and at that time their yield were very high, but due to some factors like insect pest attack, the salt tolerance and salinity of the soils, so many factors, maybe biotic or abiotic factors, that variety nowadays become outdated. That varieties are called obsolete variety. Varieties in cultivation means whatever varieties we are cultivated presently are called varieties in cultivation. Breeding lines, these are the inbred lines of a particular crop varieties. They can be used in the breeding programs. Wild forms or wild relatives are whatever wild types of variety present in the nature is called wild varieties. What is gene pool? Gene pool, you can say it is the sum total of gene of a particular crop plants. Gene pool may be primary, secondary, or tertiary. What is primary gene pool? Primary gene pool, it is we can define it as variety of the same species that can intermate freely. Means any varieties of single species which can be which can intermate and produce a fertile offspring, then it is called as primary gene pool. So crossing between primary gene pool is very easiest, and there is no any this barrier. If you talk about secondary gene pool, it is just some distance from primary gene pool. In secondary gene pool, it is closely related species that can intercross with gene pool 1 and produce at least some fertile hybrids. Not all fertile hybrids, but it can be crossed with the gene pool 1 varieties and produce some of the, suppose 100 variety 100 crossing we are doing. So in out of 100, some the 10 20 fertile offspring can be produced. So these are called secondary gene pool. So secondary gene pool is somewhat distant than primary gene pool. Then it is your tertiary gene pool. What are tertiary gene pool? Tertiary gene pools distantly related species that can intercross with gene pool 1 and 2 but requires additional measures such as embryo rescue, chromosomal doubling, this genetic engineering. So some we have to apply some genetic engineering technique to make crossing with gene pool 1 or gene pool 2. So these are the distant, very distant from the GP1 or GP2. So these are the concept of gene pool: GP1, GP2, GP3. GP1 easily cross and produce all fertile inbreds or fertile F1. Secondary gene pool means which can cross but some fertile hybrids can be produced. Tertiary means which are very distant and crossed can be crossed with the GP1 and GP2 but require some additional measures. So this is the concept of gene pool. What is germplasm collections? It is a large number of lines, varieties, and related wild species of a crop species. There are so many crop species in which we are knowing only one or two varieties. Like if we are talking about rice, only hardly 10, 5 or 10 varieties we are knowing. But if you see the germplasm of the rice. There are thousands of the varieties of the rice which are found in the nature. If we talk about maize, pulses, fruits, whatever plants we are growing, there are thousands of varieties of different lines or varieties of the different plants. So these plants varieties can be conserved at a particular place, either in situ or ex situ. In situ, either in their natural habitat or outside their natural habitat. So this collection or conservation of all varieties of a particular lines of a crop is known as germplasm collections. Like in figure, you can see this is one of the plant. It's has been preserved in the lab in test tube. This is one of the vegetable plants has been preserved. This is the vegetable plants preserved. These are flowers, bananas. This has been preserved. These are the varieties of azolla which has been preserved. So the forest in which particular plant species has been preserved. This is seed bank in which all seeds has been preserved in your banks. So this is the process of germplasm collection or germplasm conservations. So there are total notified center in all over the world in which germplasm can be conserved. Suppose if you see in North America, there is one city that is called Mexico, where CIMMYT, Maize and Rice Research, Maize and Wheat Research Institute is situated here. Maize, common bean, sweet potato, upland cotton, sesame, pepper, tomato, vanilla. This has been preserved in Mexico. If you come in Brazil or South America, here potato, common bean, peppers, groundnut, pineapple, cassava, pima, cotton, cocoa. These have been preserved here in conserved here in a seed bank. If you come in Africa, in African country like Kenya and some other countries, sorghum, teff, American rice, pearl millet, cowpea, bambara, groundnut, coffee, watermelon, these have been preserved in African countries. If you come in Central Asian country or West Asian country like Afghanistan and other country, there wheat, barley, pea, lentils, chicken, chickpea, grapevine, safflower, dry fig, date palm. It has been preserved here. If you come in India and China, that is cucumber, aubergine, banana plantation, mango, nutmeg, coconut, pigeon pea, mug, rice bean. This has been preserved. If you go in east of the China, then Asian sea, soybean, azuki bean, orange, lime, grapefruit, apricot, peach, tea, cabbage. It has been preserved. So these are some of the important centers all over the world where we are conserving germplasm of different crops depending upon the suitability of the climate as well as weather. Then what are the activities? How we are conserving the germplasm? So mainly there are four type of activities which involved in the germplasm conservations. What are these activities? It may be collections, it may be conservations, evaluation, cataloging, multiplication, and utilization. Total six steps are there. First, we have to collect the germplasm. How we can collect? Either by exploration or by procurement. Either we can search the particular germplasm in their natural habitat, or we can procure if it is not found in our country or if it not found in particular habitat, we can procure it, purchase it from some other country where it is found. So first step is collection. After collecting that germplasm, we are going for conservation. Conservation may be in situ conservation, may be ex situ conservation. In situ conservation means we are conserving the germplasm on their natural habitat, and ex situ conservation means we are conserving this germplasm outside their natural habitat, maybe in botanical garden, maybe in seed bank, maybe in gene bank. After conservation, what we are doing? We are doing evaluations. Whatever we are conserving, what are the important criteria? What are the important characters? What are the important qualities present in that conserved variety? We are evaluating depending upon their potential to use in the breeding program. After evaluation, we are doing cataloging. Cataloging means that is data storage and retrieval. We are assigning some number of the plants, like some prefix we are giving IC, EC, IW, like indigenous collections, this exotic collections, indigenous wild, like that. We are fixing some code and number so that we can use that plant in our breeding program. Then your multiplication and distribution. After cataloging, we are doing multiplication. Suppose it is required by the farmer, or it is required in the breeding program, or it is required by for some regional area variety. We can multiply and distribute. After that, we can utilize. Then it comes center of origin. Center of origin concept was given by North N.I. Vavilov. So what they have done, they have identified some particular areas all over the world in which particular crop has been originated. So total 8 center of origin is given by Vavilov. What was that? That maxi if you go in South North America, it was Mesoamerica, Andes, Mediterranean, Turkey, Iraq, Ethiopia, India, China, Southeast Asia. So these are total 8 centers of origin in which different crop variety has been origins. So generally center of origin is that the area in which maximum diversity of particular crop has been found. That is called center of origins. Then conservations, as already we have told, conservations are of two types: in situ and ex situ. In situ conservation may be by natural national park, biosphere reserve, wildlife sanctuary. Ex situ may be by seed bank, gene bank, and botanical garden. Germ plant evaluation means assessment of germ plants from various state and potentially used in plant breeding. At international level, generally IPGI, International Plant and Genetic Research Institute, Rome. It has been situated in Italy, Rome, and national level NBPGR, National Bureau of Plant Genetic Research, situated in Delhi. They are monitoring the conservation activities of all the plant varieties. Germ plant evaluation may be means each germ plant whatever we have collected, we are giving some accession number to with prefix IC. IC means indigenous collection, EC means exotic collection, and IW means indigenous wild, with variety name, their place of origin, adaptation, and its various feature. Means whatever plants we are collecting or procuring or evaluating, we are conserving or by some they are giving their basic information. Just like this is housekeeping, whatever new things we are getting, we are keeping all the data on their record, so that it can be used easily used in the breeding program. After germplasm evaluation, we are doing utilization. What we are doing of this evaluated germplasm, we can directly release apps. If we found that collected germplasm is very good and it has all the parameters at par in the breeding or in the existing varieties, we can directly release that germplasm as a varieties. Then second, if some undesirable characters found or some it is not suitable for directly releasing as a variety, we undergo for selection. We are undergoing different 2, 3, 4, 5, 6, 7, 8 selections. After selection, we are releasing that varieties. Maybe used in parent hybridization program, whatever hybridization program at national or regional level we are conducting. At that hybridization program also, this germplasm can be directly utilized. So these are the germplasm utilization techniques. So that's all about domestication, center of origins, and some introduction. In next video, we will discuss plant introduction, mode of reproduction of the flowering plants, self-incompleteness, male sterility, pure line selection, mass selection, and various selections in self and cross-pollinated crops. Before discussing that, we should know some basic terms which are used in next video. So what are basic term? That is agronomic trial. What is agronomic trial? It is the evaluation of new strain against best variety of given area and at desired parameters. Whatever desired parameters we are conducting, we have to select some best variety of particular area, and we are comparing the our new strain or new variety on from that check variety or best variety of that particular area. If new variety surpass that parameters, then we can release it as a variety. This is called agronomic trial. Genes and allele, everybody knowing gene is the functional unit of DNA. Alleles are the alternative forms of the same genes. Means suppose one gene control height, it has two forms, tall or dwarf. Tall and dwarf is called alleles. Apogamy, apomixis, and apospory. Generally, apo word is used wherever we are developing embryo without fertilization. So, if embryo is developed without fertilization from synergists or antipodal cell, then it is called apogamy. If an embryo developed without fertilization, then it is called apomixis. If embryo has been developed without from vegetative cell of ovule, then it is called apospory. F1, F2, F3, everybody is knowing. F1 is first filial generation when progeny obtained by crossing two different genotypes, then it is called F1 generation. F2 means if F1 progeny is self or undergoes selfing, then it is called F2. Further F3, F2 progeny undergoes selfing, F3, and so on, so on. Back cross, if F1 crossed with any parent, it is called back cross, and if F1 crossed with recessive parent, then it is called test cross. Double cross, we are making two different cross, P1 plus P2, further P3 plus P4. We are getting two F1 and F2. If both F1 if both F1 crossed, then it is called double cross. Emasculation, everybody knowing, removal of anther from hermaphrodite flowers. There are three types of flower: unisexual flower and bisexual flower. Bisexual flower is also called hermaphrodite flowers. So hermaphrodite flowers, if we remove their anther to avoid self-pollination, then it's called emasculation. Heritability, it is the proportion of total phenotypic variation arise due to genotype. Whatever phenotypes we are seeing, whatever physically we are characters we are seeing, how much contribute characters came directly from genotype. This is called heritability. Homozygous, heterozygous, homogeneous, heterogeneous. These four terms are always confusing. What is homozygous? This is individuals with two or more identical alleles of same genes. If two or more identical genes, alleles of same gene is present, then it is called homozygous population. If individual having dissimilar allele or different alleles of same gene is present, then it is called heterozygous. Homogeneous, individual with same genotype or phenotype, then it is called homogeneous. Heterogeneous means individuals with different gene or genotypes or phenotype, it is called heterogeneous. Means homozygous terms is used for different alleles and geneous used for different genotype or populations. Horizontal resistance and vertical resistance. Horizontal resistance is governed by polygenes, or the resistance against many diseases is called horizontal resistance. Vertical resistance means resistance against particular strain of the microorganism, that is governed by single gene or oligogenes. This is called vertical resistance. Heterosis, habit, vigor, everybody knowing. Superiority of F1 over both parent is called heterosis. Inbreeding depressions, loss in vigor due to inbreeding. Inbred lines, homozygous line developed by continuous selfing. So these are the some of the important terms which we are going to discuss in next video. So in next video we'll discuss plant introduction, mode of reproduction in flowering plants, self incompatibility and male sterility. If you have any doubt or confusion, please ask your doubt or comment this confusion in the comment box given below. I will reply within 24 hours. You can send mail also on my this given mail ID if you have any doubt. Please subscribe, share and comment so that you can get notification for later videos whenever uploaded, and others also got benefited when you share these videos. Thank you for watching this video. Have a nice day.